Hello, welcome to this introductory module to seed quality and health in the frame of a set of training materials produced by the Life Seeding Project. You can take this module on its own to just get a general overview of the topic. Or you can combine it with other modules to take one of the self-paced training paths proposed by the Life Seeding Project. My name is Stephanie Kletke and I work at ITAB, the French Organic Food and Farming Institute. I'm an agronomist by training and my current activity at ITAB is focused on the management of organic seed quality, especially in farmer seed systems and community seed banks. This video unit has 4 parts. First, we will be speaking of the components of seed quality. Then I will present physiological quality and why it's important. Then we'll talk briefly about seed microbiology and why that is important. And finally, I'll explain why we can see seed and plant health as a continuum. Let's first have a brief look at the different aspects that we need to take into account when we're talking about seed quality. Seed quality has 3 basic components. The first is specific and varietal purity. We won't focus on this one in this module. It refers to the fact that when you buy or get a packet or bag of seeds, you need to know what's inside. Specific purity means it contains the crop species indicated and not other species, for example, weed seeds. Varietal purity means that the seeds in the packet correspond to the cultivar, to the right cultivar. If you bought the tomato cultivar Red and Tasty, for example, you don't want it to be mixed up with another cultivar. This also applies to population varieties or organic heterogeneous material, even if they are not so-called pure varieties. The second aspect of seed quality is physiological. This refers to the germination capacity and vigor of seeds. We'll talk more about that in a minute. And the third aspect is microbiological. Routine seed analysis usually focuses on seed-borne plant pathogens to evaluate seed health. But we will see later in this video that seed-associated microbial communities are actually more complex than that. So, why is physiological seed quality important? Basically, the physiological quality of seed indicates its germination rate and how well or how quickly it will sprout and emerge. A simple germination test will tell you what proportion of seeds will sprout. However, if you have a closer look at seed physiology, there can be further differences between 2 seed batches, even if they both have the same germination rate. Here's a graph showing the result of an experiment with sugar beet reported by 2 researchers from Warwick University and the University of Birmingham in 2016 to illustrate this. The germination rate, as established in a lab germination test under optimal controlled conditions, is indicated in dark gray for 9 different seed lots. You can see they all have a germination rate above 95%. The same seed lots were sown under field conditions, and field emergence was evaluated, and emergence rates are indicated in light gray. Emergence in the field is lower than the lab germination rate for all of the lots, but some did much better than others. 2 have emergence rates as low as 50%, while the best performing lot emerges at about 85 or 90%. This illustrates the importance of seed vigor to ensure seeds don't only sprout under optimal conditions, but can also cope with different limiting stress factors that appear in the field under practical conditions. This is particularly relevant under organic growing conditions, where quick emergence is even more important to cope with the presence of weeds, for example. The seed emergence and seed vigor of a seed lot mainly depend on environmental factors, although plant genetics also play a role. The environment plays a role at different stages, taking into account that seeds evolve in at least 3 environments from its development to sowing. During seed development, the field conditions of the seed crop determine future seed vigor. For example, a seed crop which is well nourished will produce more vigorous seed than a seed crop which is lacking nutrients. Then, seed handling and storage affect seed vigor. 2. Seed that is stored under good conditions will preserve its germination vigor longer than otherwise. In the training modules 12 and 13, Stephen Roth goes into much more detail on the physiological processes underlying seed vigor and the key factors in seed production, which are seed harvest, seed drying, and seed storage. Finally, the last environment which affects seed and seedling vigor is the soil into which the seed is sown. Depending on the conditions and possible stress factors which happen there, a seedlot may cope and emerge, or maybe it may fail to do so if conditions are too tough. Of course, this is not something that we can control when selling or distributing seeds. Next, let's have a look at seed microbiology and why it's important. When propagating seeds, farmers also propagate the microbial communities associated with these seeds. Microbial life associated to seeds, also called the seed microbiota, has received growing attention in scientific research in the past 10 to 20 years. Scientific research has shown that seeds and plants in general have strong interactions with their microbial communities and that microbes affect many plant physiological processes. Plants are therefore considered as holobionts. That means they are living entities composed both of plant and microbial cells. This also goes for seeds. Beneficial effects of some microbial species associated with seeds have been demonstrated by scientific research, but for most species, functions are still unknown, or they depend on environmental conditions and interactions. That means they can sometimes be beneficial, sometimes be detrimental, or often be neutral. Some seed-transmitted microbes are also potential plant pathogens. I call them potential pathogens because the appearance of disease symptoms always depend on environmental conditions that are favorable to the disease. However, if such microorganisms are present in a seedlot, this comes with the risk of plant disease in the following crop. You are maybe wondering how these seed microbes can be beneficial for the seed and the seedling. Firstly, some species have been shown to make an active contribution to seed conservation, thereby improving seed storage for a longer shelf life of seeds. They can also facilitate the germination process and stimulate plant growth and plant defenses. When sown, the seed microbiota can be a protection against diseases and especially soil-borne plant diseases, especially in soils with poor soil life. Here are two examples from science that demonstrate a role of plant and seed microbiota for organic plant breeding and seed propagation. Firstly, in an experiment conducted by Purdue University in collaboration with the Organic Seed Alliance in the USA with carrots, microbial species present on taproots were analyzed. Organic cropping systems were shown to favor microbial species that mitigate disease caused by Alternaria daudzi. Carrot cultivar also had an effect on microbial composition. Some microbial species favored by organic systems were shown to favor seed germination, seedling growth, and tolerance to Alternaria daudzi when inoculated on seeds too. Authors conclude that carrot breeders could potentially begin to select for these beneficial plant microbial relationships. Results of another experiment with tomato at Graz University of Technology indicate the seed microbiota can be explained by an adaptation to prevalent environmental conditions. Seeds generated under field conditions in this experiment showed traces of a bacterial community composition connected to the suppression of plant pathogens. Seeds of the successive generation, grown in a pathogen-free and low-nutrient environment, showed a predominance of bacteria that facilitate the uptake of nutrients. In this experiment and with this crop, seeds were identified as a primary primary vehicle in the transmission of plant growth promoting bacteria. Authors of this study conclude that this constitutes a strategy to maintain plant beneficial bacteria over generations, and that this should be taken into account when designing seed treatments. These are just two specific examples. Their results should not be generalized to all crops and contexts. They do represent promising perspectives, although we are still far from practical recommendations for organic seed production. To learn more about seed microbiota, their functions and assembly, take Module 14 with my colleague Gaspard de Tournemire. It is important to keep in mind seeds also bear microbial species which can cause severe plant disease if they find the right environmental conditions. On cereals, for example, both common bunt on wheat and loose smut on barley are seed transmitted, requiring caution when propagating and distributing seed. Prevention is even more important in organic seed systems because the use of synthetic chemical fungicides as seed treatments, as in general, are refrained from. Module 18 addresses the specific case of breeding strategies to control common bunt and associated precautions in seed propagation of common bunt-resistant varieties. It is based on documents produced by the biodynamic breeder Dottenfelder Hof in Germany. Here are two more examples of seed-transmitted plant diseases in vegetables. On the left, you see a symptom of bacterial canker on tomato fruits, caused by a bacteria that is transmitted from seeds to the following generations of tomato plants. On the left is a picture of leaf symptoms of seed-transmitted bean common mosaic virus on common bean. I hope these considerations and examples have made it clear that plant and seed health are really a continuum. This is the last aspect I want to address in this video. Indeed, I'd like to stress that seed quality is affected all through the growing cycle, as represented here in a diagram designed by Wageningen University and Research in the Netherlands and ITAB in France as part of the LifeSeed project. Healthy, well-nourished seed crops will give healthy, vigorous seeds, provided that seed harvest practices are adequate and that care is taken during seed processing and storage. When seed has good vigor, seedlings will emerge quickly and be more tolerant to various environmental stress factors, which is the best basis for successful organic crops. By providing good quality, healthy organic seeds, seed companies, farmer seed networks, and community seed banks contribute to productive and resilient crops in organic fields and gardens. The plant and seed microbiota play a role in this process, as microbial species can favor plant growth and health as early as in the seedling phase. Much further research is required to really understand interactions between crop plants and their microbial communities, especially under field conditions in organic systems. Of course, pathogenic microbial species among the seed microbiota require special attention and care. To wrap up this video, let's summarize some of the key take-home messages. Firstly, let me remind you that the three main components of seed quality are specific and varietal purity, physiological quality, which affect germination, and microbiological quality that affect seed health. Secondly, the performance of seed is determined all along the propagation cycle and determined by environmental factors during seed propagation in the field, during seed handling and storage, and finally, sowing conditions. Thirdly, seeds carry microbial communities and interact with them. Effects can be beneficial, neutral, or pathogenic. Managing seed-transmitted pathogens is important in organic seed production. I hope you enjoyed this video unit. In the following one, I will talk about the factors underlying seed quality and health, and about how the different aspects are actually intertwined with each other. I will also give some examples of critical situations for seed quality in organic seed propagation and distribution. Thank you for your attention.